All right, I just want to get started with a few parts of chapter 4, just to get us going, and then we'll call it a day. Okay. I'm imagining that on Thursday you're going to have lots of questions about what's going to be on the test next week, because you'll have done your homework by then, but we'll see. So let's talk a little bit about chapter 4. So chapter 4 is titled Extensions to Mendelian Genetics. So these are different fractions or different ratios, still due to probability, but there are genes that behave in a different manner than what we call completely dominant genes. And always the first extension we start with is incomplete dominance. And again, those of you that maybe learned about this in Bio 1, will just be a quick review. So the first extension, incomplete. It is like it sounds. It means that the dominant allele does not completely mask the recessive allele. I'll say that again. The dominant allele does not completely mask the recessive allele. Not now. So the F1 generation is a blend of characteristics, a blended color, and in between, a different leaf size, a different color, a different shape, maybe. The classic example is because it was the first one that was observed, is flowers, snapdragons. Red snapdragons were crossed with white snapdragons. No one knew what the dominant phenotype was going to be. Most other things we expect the color versus is usually dominant versus the absence of color. So I think the scientists were predicting the flowers were going to be red, but they weren't red, they were pink. All the F1 was pink. And this happened actually like right when Mendel's work was being rediscovered. So there were a lot of people that said, oh, Mendel was wrong. There is blending inheritance. However, when those pink flowers were allowed to self-fertilize, we see the reappearance of the white flower in the F2 generation, just at a different proportion. So we red flowers, pink flowers, and white flowers. So let's go through and assign those symbols and talk about how that's still Mendelian genetics. And I'm just checking because I want to use the symbols that are in your book. He's using R1 for red and R2 for white. So R1 would be a true breeder or a parent generation for red, crossed with R2, which is a true breeder in this case for white. So I think without me completing the Punnett square, most of you can do this now, that the F1 are all going to be heterozygous. And as I mentioned, rather than them being red, which is what we would normally expect, they are actually all pink. So R1, R2 genotype translates to a pink phenotype. And again, when this happened, the first assumption was, oh no, Mendel was all wrong. There is blended inheritance. They're a blended color. They're an in-between color. However, it turns out when you let these flowers self-fertilize, the gametes, the chromosomes still assort into the gametes or segregate into the gametes randomly with equal frequency. So when we have an R1, R2, crossed with an R1, R2, so two pink flowers. Okay, so do it with me, right? R1, R1. What color is that one going to be? That one's red, correct? So I have 1/4 that are red. R1, R2, what color are those going to be? Those are pink. How many do I have? What fraction are pink? 1/2, right? And then R2, R2, that homozygous recessive shows up again in what percentage? 1/4, yep. 1/4 white. There are many traits that are incompletely dominant, and they're like, oh, what can I say? There are many traits that are incompletely dominant. How would I know in a genetics problem if a trait was incompletely dominant? I would have to be told, or I would have to be told the parents and see that the F1 generation is a different phenotype, right? So let me say that again. How would I recognize if a trait is incompletely dominant? I would have to be told, right? In grasshoppers or in frogs, green is dominant to spotted green with brown spots. A green frog crossed with a spotted brown frog gives me a green frog with spots. This is an incompletely dominant trait. So I would have to either have a new phenotype listed in the given part of the problem, or I would have to be told specifically, you know, spotting in frogs is incompletely dominant. Green is incompletely dominant to brown with spots or whatever. Maybe that wasn't a good example, but I think you get the idea. And again, if I was asked to do a chi-square test with an incompletely dominant problem, do you see how now I would be multiplying by different fractions for my observed numbers, for my expected numbers, right? It would not be 3/4 and 1/4. It would be 1/4, 1/2, and 1/4. So my chi-square table would get bigger. And how many degrees of freedom would I have in this case? 2, right? Because I have 3 phenotypes. So n-1, 3 minus 1 is 2. Yes. Are there situations where you have a dihybrid, you're looking at 2 traits and they are both incompletely dominant? Absolutely. These extensions, what we're going to see is that in the real world, obviously, even Mendelian traits are a lot more complex than we originally thought, or maybe we originally knew. Question so far? You guys heard that before? Yes, no, maybe so. Yes, no. You're okay with it? But wait, there's more. All right, so incomplete dominance, codominance. Codominance is a situation where both alleles are dominant. So we have 2 alleles and they are both dominant. We're going to see different versions of expression, right? We may see different proteins, for example. The example we normally give is the M and N. Define it first. Both alleles are dominant and appear in the phenotype. And again, the example is usually M and N. These are either proteins or carbohydrates on blood cells. I think they're on red blood cells, separate from A, B, and O. So there are still two alleles, but notice that I've made them both capitals, not a big one and a lowercase one for the same, and it's one trait, it's one characteristic, but you either have, and you get two copies, one from each parent, but again, you could be one of three phenotypes. You could be MM, you could be MN, or you could be NN. One protein, different versions, or one carbohydrate, different versions. We always use this to lead up to talking about human blood groups because it is multiple alleles as example of multiple alleles and codominance, right? When Mendel was studying the peas, everything had two versions or two alleles, two flavors, two types, right? You were tall, dwarf, round, wrinkled, yellow, green, purple, white, right? Two choices. What we know now is for a lot of characteristics, it's more than three alleles, or more than two alleles. It could be three, it could be four. Each individual organism is only going to inherit two, one from each parent, but in the gene pool, there are more than two choices. Probably a good example of multiple alleles that helps we can use among people, humans, right? So for example, skin color in humans is controlled by 30 genes. Each of those genes has at least two alleles. Just think about that for a minute, right? So I'm inheriting one copy of 30 different genes, but I'm getting all different variations or all multiple alleles of those genes. So even with siblings, right, you often are not literally exactly the same color because you've inherited 30 different genes and multiple alleles of some of those genes. What? It's crazy. Right? Let's look at blood type because that's an easy one. It only has three alleles. A, B, right? And these are actually the phenotypes, A, B, and O. We genetically indicate them with an I with a superscript of A. And O is actually depicted like that. A and B are co-dominant, but we have three alleles. Multiple alleles, right? So an individual could inherit two copies of the A allele, and their blood type would be A. They could inherit one copy of the A allele and one copy of the recessive allele O. Their blood type would still be A. They could inherit from their parents one of each of the dominant alleles. They would be blood type AB. They could inherit two copies of B, right? One copy of B and one copy of the recessive, or they could inherit two copies of the recessive trait. Then what blood type would they be? Oh, right. So they could be blood type A, blood type AB, blood type B, or blood type O. Right, so we end up with four phenotypes with those three alleles. O is recessive, A and B are co-dominant. If you have both of them, they are both expressed on the surface of your red blood cells up. Incomplete, multiple. Flip the page here. Ooh, lethal. That's cool. Let's talk about lethals. And your book puts the specs links at the end, so I'll try to stay in line with what they do. Number 4. Lethal genes. These are the one that I can think of is one that is a lethal recessive, I think. And if you have it, 2 copies of it, that offspring is never born. Okay, because it's an essential gene. So if you have 2 copies of the mutant, it's not a viable offspring. It's usually in mice. We've got yellow and agouti. So agouti is the normal mouse color, brown, basically. But if you actually pulled out a hair of that mouse, you would see, like on your pets, there's areas of the hair shaft that are different colors, but combined from a distance, it looks brown. And let's see, it's a recessive lethal allele, and it is a homozygous recessive. So if you'll never see any homozygous recessive for yellow mice. There'll be heterozygotes, but that mouse doesn't survive. Mice with the genotype A yellow, don't get them. All right, so I want to do a few more with you, but not, again, some of these, like this one, like the codominance and the incomplete dominance, I'm going to assign you some problems for those, probably assign them on Thursday, because you have enough problems to work on right now from chapter 3. The next set, the epistatic problems, I wouldn't assign those for you to actually work out the problems. I would expect you to know the definitions of epistasis and the epistatic gene and the hypostatic gene. And I would expect you to be able to recognize a dominant epistasis versus a recessive, meaning in sets of the phenotypes. And I'll show you, there's a table in your book you can look at, but I'm not going to ask you to solve particular problems for those, like chi-square problems. Okay. I know, you're just looking at me like, what is she talking about? So this is when more than one gene influences a single trait or characteristic. So it's 5. Sometimes you'll hear it referred to as gene interactions because one gene or one character, one protein, one enzyme is going to control the expression of another one. And it's in the same characteristic or trait, right? So one gene controls the expression of a second gene. And we're going to look at coat color in dogs, coat color in Labrador retrievers, because it's something that we're all familiar with. But I'm just going to give you the definitions real quick of the hypostatic gene and the epistatic gene before we go on. Sorry about that. Oh, it's double star Tuesday for me. I'm gonna have to go to Starbucks. All right. Because I always forget, so I'm looking. There we go. So in this case, the alleles at the first locus are epistatics at the second. So where's my definition? So why can't I find that? All right, I thought I had everything on. All right, well, that's not that important. So we're going to look at coat color in dogs. Like I said, we're going to look particularly at Labrador Retrievers. And this is not a picture from your textbook. This is a picture from your Bio 1 book. Small. Move me. Here. Turn that into that. There. Can you see it? Yeah. Okay. So we've got two genes here, the B gene and the E gene. Okay. Big B and big E. So what we're looking at is two heterozygous dogs for those two genes. If you notice, they are both black labs up at the top, right? So we have black lab that is big B, little b, black lab that is big E, little e. Really, they had to both sound the same. That doesn't make it easy. Big B, little b, big E, little e. So they're heterozygous for both of these traits, right? What we would expect, because we've been doing this a little while now, we would expect in the F2 generation to see 9:3 to 3:1, right? But if we look at these offspring from, it's done based on a pedigree analysis, lots and lots of puppies, we see that we have 9 black ones, 3 brown ones, and 4 yellow ones. We only have 3 phenotypes, not 4. We only have 3 phenotypes in this case. And one of them appears to be like a combined genotype phenotype, right? We don't have another color, and something is changing that one into yellow. Now, I want you to look at the genotypes of all the animals and their colors, right? And tell me if you can find the pattern. And which gene is control. I'll tell you which one's controlling which after you figure it out. What do I need? What does the dog have to have if it's yellow? 2 small E's. So no matter what I have at the B gene, if I have 2 little E's, it's going to be a yellow lab. True? Is there any other dog that has 2 lowercase E's and is a different color? Nope, all the yellow ones have 2 little E's. So the E gene is the controller, right? So what that means is if I have 2 little E's, the pigment that's either going to be brown or black is blocked from being deposited into the fur. So it doesn't matter if I'm making brown or black pigment, or if I can make brown or black pigment, it can't be put into the fur. So that dog is going to be yellow. As long as I have 1 capital E, then it matters what I have at the B gene, right? Which is dominant, black or brown? Black, right? Because we've got 9 black ones, right? 3 brown ones. So 9 must be black, or yeah, black must be the dominant one. So if as long as I have 1 capital B, it doesn't matter what I have as my second trait there, and 1 capital E, I'm going to get a black dog. Right? The only way I get a chocolate dog, the only way I get a brown dog is the recessive to the B gene. I have 2 copies of little b. I still have to have 1 big E so that brown pigment actually gets deposited into the hair. Right? And again, we have these at 9:3 to 4. That is an example of recessive epistasis because the recessive is the E gene is the controlling gene, and the recessive form of it is controlling the expression. The expression in the phenotypes, if it's a dominant epistatic event, I believe, or dominant epistatic genes, I believe that's 12 to 3 to 1. Yes, write that down. Dominant epistasis, the modified ratio would be 12 to 3 to 1. We still only have 3 phenotypes. But in dominant epistasis, what's going to happen would be the chocolate and the blacks would be merged, and we would still get the other 2 phenotypes. Right? Plants, not so much in dogs.
Recessive always 9, 3, 4.
If it's recessive epistasis, it's 9, 3, 4 if there's no other factors involved. Let's just get crazy for a minute. And what if I said the gene for the depositing of the pigment, if it's incompletely dominant, right? Or if the gene, or if there are multiple alleles for the color, right? So it gets a lot more complicated really quickly, but yes, for our purposes, that was just to make your brain hurt, right? For our purposes, 9, 3, 4 means recessive epistasis. 12, what did I tell you? 12, 3, 1 is dominant epistasis because it's a combination of the dominant phenotypes rather than the recessive phenotypes. All right. Any quick questions or simple questions about that right away? No? All right. Then let's carry on. One more on sort of kind of on this list. 6. It's called pleiotropy, and I don't want to spell it incorrectly. Sorry, yeah, tropy, not trophy. Pleiotropy. And this is, I like to think of it as sort of the opposite effect. This is when a single gene affects multiple parts of a phenotype. I usually use the example of sickle trait because it not only affects the shape of the red blood cell, but it can also affect the spleen, lungs, heart. I believe your textbook gives the example of Marfan syndrome, right? A human malady resulting from an autosomal dominant mutation that codes for the connective tissue protein fibrillin. Because this is widespread in many parts of your body, you can imagine it has effects all over the place, but it's a single mutation in one gene. So that's different than epistasis, where one gene is controlling the expression of another, and it's just showing in one part of the phenotype. This is one gene showing in many parts of the phenotype. All right. And we can add sex-linked traits here as well. So as we mentioned when we were talking about pedigrees, a sex-linked trait is a gene that is located on the X chromosome. I'm going to put in parentheses here in humans and fruit flies. Because in other animals, fish, some birds, some insects, sometimes the sex characteristic is the chromosome is not X, it's Z or W, depending on the organism. Because these genes are sex linked, because they are located on the X chromosome, they can only, if they're a recessive mutation, if you have one copy, so I'll put recessive mutation, one copy. So in humans, if you're male or if you're a male fruit fly and you have one copy of the characteristic, the trait, the mutation that's on X, you will see it in the phenotype. That's why we say you see the expression of those in pedigrees more frequently in males than in females. And we were so careful to say the autosomals will see equal expression in males and females. It's going to be much more rare to see a female with a sex-linked trait because she's going to have two copies of that sex-linked trait in order for it to be expressed. So 2 copies of the mutation. To be seen in, I should put phenotype of a male here, to be seen in phenotype of a female. In humans, there's a really short list of sex-linked traits. I believe they have them in your book. They were first identified in fruit flies. The white-eyed fly was the first ever mutation that was linked to a particular chromosome. It's linked to the X chromosome, which happens to be the sex chromosome in fruit flies as well. The mutation was seen in a male fly, and then when it was crossed with female flies, in order to get a female fly that has white eyes, you have to have a female carrier parent and a male that is white-eyed, right? And then you can get white-eyed female. So that cross is pictured in your book on page 79. I thought they had a list. Here it is. There's a list in your book of human sex-linked traits or X-linked traits, color blindness, a G6 deficiency, glucose 6-phosphate dehydrogenase deficiency, 2 types of hemophilia, Lesch-Nyhan syndrome, and Duchenne's muscular dystrophy. That's it. So we got 5. No, but if they show up in a problem, right, then if you have some idea of what they are, then then you're like, oh, that's a sex-linked trait. But if you don't know that right away, but like, for example, if I ask you to draw a pedigree, I would have to tell you it's a sex-linked trait, right? Or if I gave you a probability problem to solve, I might say, color blindness in humans is a sex-linked trait or is an X-linked trait, so that you would know. But it just helps, if you know what they are. Again, those are more rare in females because you have to have 2 copies of the trait. Duchenne's muscular dystrophy is people usually don't live to past the age 20, and Lesch-Nyhan, I think they only live not very long at all. Hemophilia you can live with. I think you can live with G6 PD phosphate dehydrogenase. You can live with that. You just have to moderate what you eat, and of course you can live being colorblind, right? You might just be a bad dresser, but it happens. I knew once upon a time, I knew a woman who was colorblind, who married a colorblind man. I'm not kidding. And all their children were colorblind, right? It's very on the mild side, but she thought she had brown hair. And no, she had red hair. Like she had orange, like auburn, strawberry blonde hair, like really like orange hair. And she came to work one day and she said, I said, oh, I like the hair color. Did you color your hair? She's like, oh yeah. It looks so much better toned down brown like this. And I was just like, it's orange. I said, really? She's like, yeah. And she had pictures of her children on her desk. And I was like, what color is Brianna's hair? And she was just like, oh, it's a light brown. And I'm like, what about Brendan? She was like, oh, he has brown hair too. And I'm like, what about Steve? And she's like, and Steve actually did have brown hair. She's like, oh, he has brown hair. And I was like, what color hair does Steve think is it? He's like, he thinks it's brown. I'm like, is he just saying that? She's like, no. We picked out the color on the box. We went to the drugstore and we picked it out and we did it at home. And I was just like, so you're all colorblind. Yeah, all of them. But super rare to meet a colorblind woman, right? All of us have probably known colorblind males. It's a lot more common, right? Siblings, just friends.
My boyfriend.
Your boyfriend's color, like for real? Blue.
No, just like, yeah, like blue, purple, like dark colors like that.
Yeah. Yeah, there's all different versions of it.
Yeah.
The most severe being red, green color blindness, right? That's why stoplights, that's why on your driver's test, they ask you which position of the traffic light means stop. They're not trying to trick you. It's so colorblind people can drive. You're supposed to say, the one at the top, right, is red because colorblind people can't distinguish very easily from the red from the green, right? So. All right, sex limited and sex influenced. Those are the last two, I think, that we're going to talk about today anyway, and then we'll finish off this chapter on Thursday with expressivity and penetrance. So sex limited and sex influenced. They're related to the sex linked. So the sex influenced and sex limited are like they sound, right? So if the expression is limited to one or the other sex, in the case of sex limited, only males show the phenotype. And your textbook gives the example of feathering in chickens. So let's take a look at that. Here's some chickens, a hen and a cock. Okay. So we have, again, it's this button. Not great, but best we can do. Like I said, you can see them in your textbook. So it has to do with the feathering patterns, tails, obviously, right? And the females can have the trait, but they're never going to express it because it's limited only to males. It's usually a hormone controlled trait. Sex influenced, females will express it, but to a lesser degree. Like horns in some animals is sex influenced. So it's more likely to be seen in males, but you'll see it in females, but just to a lesser degree. Male patterned baldness in humans is a sex influence trait. You can see it in females, but it's usually expressed later and not as severe. It's expressed more severely in males. So just like it sounds, right? Sex limited, you only see the expression in one sex or the other. Sex influenced, you can see expression in both sexes, but in one sex, it's more like bigger, larger, different colors than in the other sex. All right, we will stop there for today.